Mitosis helps replace cells that are constantly being damaged. In prophase, single chromosomes condense into pair of sister chromatids. The nucleoli disappears along with the nuclear membrane. Prophase results in the migration of centrioles to opposite poles. In metaphase, chromosomes take position on the metaphase plate in a linear fashion. Centromeres, which are proteins that bind sister chromatids together, become connected to spindle microtubules. Anaphase is a pulling apart of sister chromatids to make individual chromosomes. Spindles that have attached to a centromere of each chromatid are pulled by each centriole from opposite poles, which result in twice as many chromosomes. In telophase, chromosomes, formerly sister chromatids, have now gathered at opposite poles. They have what is known as a cleavage furrow, which results from specialized protein pinching the cell at the metaphase plate to form two identical daughter cells. This is also known as cytokinesis, the division of the cytoplasm. Also, the nucleoli and the nuclear membrane have reformed in this stage. Chromosomes in the two daughter cells will be identical to that of the parent cell in this stage as well.
Meiosis I is important for gametic reproduction for sexual reproduction in animals. Meiosis I requires 23 chromosomes from the mother in orange and 23 from the father in blue. After moving through G1, S, and G2 of interphase, the non-sister chromatids pair up to form pairs of non-homologous sister chromatids, or tetrads, through synapsis during prophase I. Simultaneously, the nuclear envelope disappears and the mitotic spindle begins to form, with the centrosomes moving to opposite sides of the cell. In metaphase I, the microtubules connect to the kinetochores, represented by red dots, of the centromeres of the homologous chromosome pairs. As the homologous chromosome pairs randomly line up on the metaphase plate, they exchange genetic information by crossing over at the chiasmata point. In anaphase I, the homologous chromosome pairs are separated with their new genetic information attached, represented by orange and blue dots, but the sister chromatids remain attached. In telophase I, the mitotic spindle retracts and the nuclear envelope begins to reappear. Cytokinesis then uses a cleavage furrow to fully pinch off the cytoplasm and separate the cells. The resulting cells each have genetically unique information. In meiosis II, the process is the same as meiosis I, except sister chromatids are separated instead of homologous chromosome pairs. Because these chromatids still contain genetic information from crossing over, the resulting chromosomes are genetically unique as well. Because we started with 2 cells from meiosis I, the pinching of the cleavage furrow and cytokinesis in meiosis II creates 4 daughter cells. Overall, we started with 1 diploid cell with genetically identical chromosomes, and through meiosis I created 2 haploid cells with genetically unique chromosomes. Through meiosis II, we then created 4 haploid cells with genetically unique chromosomes. From our parent cell, we created 4 times the number of cells with half the number of chromosomes in each cell. Mitosis creates double the number of cells from the parent cell with genetically identical chromosomes to the parent cell, and meiosis creates 4 times the number of cells with genetically unique chromosomes to the parent cell. Additionally, in mitosis, the number of chromosomes in the resulting cells is the same as the parent cells, where in meiosis it's half